Nupur Biswas, PhD. Nupur Biswas is working as a senior bioinformatics scientist at Renix. She is working on bioinformatics and other multiple projects as well. Some of her projects include the designing personalized neoantigen vaccines for cancer patients and exploring the genes behind chordoma and the health applications of digital healthcare wearables. Nupul has a doctoral degree in physics and her research work focused on studies of confined polymers. Before joining Renix, she worked at the Indian Institute of Science, Raman Research Institute, Collaboratory, and the Indian Institute of Chemical Biology. At Renix, she has gained experience as not only a research mentor but as a scientist contributing to various international publications. So, I request Nupur ma'am to continue the presentation.
Thanks, Professor, for your introduction. Welcome, everybody. Today, I am going to discuss a paper which is titled Increased Hyaluronic by Naked Mole Rat Has to Improve Cell Span in Mice. This paper is authored by a group from Rochester University with the first author, Chiu-Hui Chang. It was published in the journal Nature in this year. It is focused on the longevity. It tells that the gene transfer extends the lifespan of mice. Although this research article is primarily focused on the longevity, it has impact on the inflammatory disorders. Now, people want to live a longer life. Achievement of long life is desired for ages. But as Martin Luther King Jr. once said, that it's the quality, not the longevity of one's life is what is important. The longer life, that's why longer lifespan must be accompanied by healthy lifespan so that people remain active. That's why enhancing longevity is an active area of research at this moment, and the focus is set on identifying the causes and the hindrance of longevity and then translate it to the benefit of the mankind. This research article is focused on main established fact on naked mole rats. Naked mole rats have an exceptional longevity. They can live more than 40 years. They are protected from multiple age-related diseases, which includes neurodegeneration, cardiovascular disease, arthritis, and even cancer. With time, they have evolved anti-aging and anti-cancer defenses. And these features are related to the presence of hyaluronic acid, which is obtained in a large amount in the mole rat tissues from different organs. Hyaluronic acid is an anionic polymer, which length can vary from one oligomer to the millions of daltons, and its properties, its activity depends on its molecular weight also. Low molecular weight hyaluronic acid is associated with inflammation and tissue injury and cancer metastasis. This research is primarily focused on high molecular weight hyaluronic acid, which will be abbreviated as HMW-HA henceforth. This high molecular weight molecules help to improve the tissue homeostasis. It shows anti-inflammatory and antioxidant properties. This hyaluronic acid is synthesized by hyaluronan synthesis proteins. For vertebrates, there are primarily three types: Has1, Has2, Has3. The current research is focused on Has2 gene. Since it is established that HMW-HA is responsible for cancer resistance in the naked mole rat, the challenge is to translate this to other species and test its role in the context of longevity, in the context of healthy longevity. So the objective of this research is set to test whether anti-cancer and potential anti-aging effects of HMW-HA can be recapitulated in species other than the naked mole rat. To explore this objective, they focused on the mouse. They generated a mouse model which overexpressed naked mole rat Has2 gene. To overexpress the Has2 gene, they choose the CRE-ER mouse lines. CRE protein is a site-specific DNA recombinase that can catalyze the recombination process of DNA, and it is used to get tissue-specific mutant mouse models. So they use CRE-ER to generate the mouse model, which are HA gene is overexpressed. Henceforth, we are calling this mouse model as NMR HS2, and the control mouse is the only CRE ER. They further use different experimental techniques, including RNA-seq DNA methylation. Coming to the results, they first check the presence of HMW-HA in the mouse tissues. Here we see in the tissues of the muscle, in intestine, the presence of HMW-HA is quite high, right? And the molecular weight is also in the high range. Whereas in the sites which are known as the breakdown sites like liver, spleen, the presence of HMW-HA is quite low. That it establishes the fact that they have successfully generated the mouse model where the HMW-HA is present in a large amount compared to its control version. But even in that case also, this amount is less compared to the presence of HMW-HA in the case of naked mole rats. But whatever the amount of the present, the effect of cancer resistance is evident in case of the mouse model. They report that 57% in NMR-H2 mice died of cancer, whereas in case of the control, almost 70% died. And the effect is more in case of the old mice, where we can see the black bar shows that 80% of the controlled mice died of cancer, whereas only 50% of the NMR-H2 mice died of cancer. And also the presence of NMR has present of hyaluronic acid is visible in the skin, both for in case of the female and the male. And the present is also quantified in terms of the measuring the intensity of the dye used to tag the hyaluronic acid. And the NMR-H2 mice also showed the increased lifespan. They have increased the median lifespan by 4%, but the maximum lifespan increased by 12%. But there was no change in the molecular, in their body weight. So it establishes the fact that NMR-H2 mice has the increased lifespan along with the presence of the hyaluronic acid in their different tissues. But to explore the biological age, they check the methylation age of the CRE mouse and NMR-H2 mouse. CRE mouse was close to their chronological age, but NMR-H2 mouse shows the decrease in the acceleration. That means they have a younger biological age. They further check the methylation age by exploring the CPG sites. It is known that around more than 6,000 CPG sites are there which show changes in the methylation while the species age. Among them, 165 sites are differentially methylated between NMR-H2 mice and CRE mice. 145 sites which are known to gain methylation during aging has showed lower methylation in case of the NMR-H2, and 20 sites which are known to lose methylation during aging showed higher methylation in case of the NMR-H2 mice. So it means NMR-H2 mice methylation is following the reverse trend. So they are exhibiting younger biological age, although chronological age is increasing. And the increased lifespan when lowering biological age is also associated with the improved health span. To quantify the health quality, they used frailty index. This index considers different parameters like body weight, hip strength, mobility, and this score increases with age. But in case of NMR-H2 mice, in case of the old NMR-H2 mice, FI score was substantially lower, as we can see here from the red color bar. Other parameters which usually are considered while as the health parameters while the disease age are also measured in case of the NMR-H2. They measure the 4-limb state, which were also quite good compared to the young NMR-H2, whereas the old control mice is dropped. They've also checked the connectivity densities of different bones like femur, tibia, and it was also quite good for enema-Has2 mice. It means this enema-Has2 mice have the possibility of getting osteoporosis is quite low. So overall, they have an improved health span. Now, to explore the inflammation of the enema-Has2 mice, they check the RNA-seq analysis. Enema-Has2 mice showed fewer transcriptome changes with aging. Aging and the transcriptome of the enema, that means that their transcriptome is less perturbed with age. Age for different tissue sites. And they further explored the role of the inflammatory related genes, and they observed reduced expression of inflammatory related genes and higher expression of genes involved in case of normal liver functions like nutrient metabolism. Now the question remains, is this anti-inflammatory effect is due to the hyaluronic acid? To explore this thing, they check the macrophage cells. Macrophage cells are the part of the immune system. They used macrophage cells and overexpressed macrophage cells by using mouse Has2 and also enema-derived Has2. And they observed whatever the source of the Has2, they both show lower levels of pro-inflammatory cytokines like IL-1 beta and higher levels of anti-inflammatory cytokines. It implies that anti-inflammatory effect arose from the production of the high molecular weight hyaluronic acid. Coming to the discussion, in the tissues of the naked mole rat, we observed that HMW-HA is abundant and contributes to the cancer resistance and possibly longevity. And this evolution adapter, which was unique to the naked mole rat, can be exported to other species. Enema-Has2 mice accumulated HMW-HA in multiple other organs, and they also show resistance to the spontaneous and chemical-induced cancer, showed an extended medium and maximum lifespan, displayed improved lifespan, acquired lower methylation age, and transcriptome had more youthful feature. Transcriptome is less perturbed while they age. So the presence of HMW is leading to a longer-lived species. But the question is, what is the molecular mechanism by which this hyaluronic acid is extending the mouse lifespan and also giving a healthy life? The answer is still not clear to the researchers. They think that due to high conservation of Has2 gene in the naked, the sequence part is not important. Actually, the important thing is the production of HMW-HA. This is a very important statement. Is the production of HMW-HA is important. So the production can be done by in situ also by using supplementary training. That's why the enema they reach to this conclusion based on supportive information that they observed, which include that enema-Has2 mice show down regulation of multiple inflammatory related pathways. They reduced inflammatory signal and improved glucose homeostasis, and also plasma of enema displayed also reduced level of pro-inflammatory cytokine. That means there exists possibly some systemic anti-inflammatory effect of HMW-HA, which needs to be explored more. And this systemic effect is leading to the benefits to those tissues also where HA is not present or not elevated. So the benefit of the elevated HA in one tissue is being obtained by the other tissues of the body also. Now the lifespan observed in enema-Has2 mice was increased significantly, but the fact is the health span actually improved more robust. And enema-Has2 mice accumulation was modest, but still it was far below than the amount of hyaluronic acid we observed in the naked mole rat. The reason can be that while generating the enema-Has2 mouse lines, they used hyaluronic acid synthesis process. They changed the hyaluronic acid synthesis process, but did not change the degradation process. In the naked mole rat, high level is associated by both robust synthesis and very slow degradation of hyaluronic acid. Now, In case of the mice model also, if simultaneously hyaluronic regression can be attenuated, then possibly more better result, more greater lifespan extension and healthy NMR has to can be achieved. So finally, we can conclude from this research that HMW-HA produced by NMR has to extends lifespan and improves health span of mice. It suppresses the production of pro-inflammatory factors. The anti-inflammatory properties of HMW-HA may potentially reduce the chronic inflammation during aging and prevent cancer initiation. The impact of the study is that this evolutionary adaptation found in long-lived species can be exported and adapted to benefit of human health, and HMW-HA may be useful for treatment of age-related inflammation in the intestine and other tissues. That's why I want to present today. If you are further interested, you can write me at nupur@renix.org. Thanks to everybody for your attention.
Now the session is open for QA. So the question from Vijay is that anti-cancer or anti-aging effect of overexpressed hyaluronic
Hello.
acid, yeah, is correlational or causational?
They actually induced cancer in the NMR mice and then observed their how many are actually living or how many died. In this way, they established the fact that presence of hyaluronic acid is reducing the cancer rate.
Yeah, the another question from Yogita. Are there other factors beyond hyaluronic that contribute to the longevity and the health span of naked mole rats?
Till now, it is the hyaluronic acid that is considered as the major factor for the naked mole rat.
The other one from Yogita is that does the function of the immune system or the gut microbiota improve with higher hyaluronic?
Yeah. It is also actually improved from the effect of the higher molecular hyaluronic acid. Yeah. Yes.
Uh, I hope those were clear. So the next presentation is on sex and age dependent genetics of longevity in heterogeneous mouse population. The presenter is Vijay Padul and on the 15th November of 2023. Thank you.